Hello everyone, my name is Anna and I'll be your host for today. Welcome and thank you for taking part in this MDPI webinar entitled Frontiers in Nucleic Acid Chemistry in memory of Professor Enrique Pedroso for his outstanding contribution to nucleic acid chemistry. This is the 28th webinar in the Molecules series based on a special issue from our journal of the same name. So if you are interested in contributing to this topic, check out the webinar's webpage for publication opportunities. I would like to remind everyone that this webinar is being recorded and that the recording will be made available as soon as possible on the webinar webpage. Today's session is also being live streamed on MDPI's YouTube channel, so hello to those of you watching us through there. I'll be sharing the YouTube link on the chat in a few minutes to anyone that could be interested to watch it through there too. Certificates of attendance are available upon request, so to get yours, please send us an email to molecules.webinar@mdpi.com. Don't worry, I will be sharing also this email address on the chat in a few moments. Those certificates will be delivered to those who have registered and are watching us on Zoom, but not on YouTube. So thank you for your understanding. This webinar will be composed of brief introductions by today's chair, Professor Ramon Eritja, and 3 lectures of 30 minutes each from today's speakers. At the end of the webinar, we will have a question time moderated by the chair. So if you have any question or comment, please post them in the Q&A box that you will find at the bottom of your screen next to the chat. Please remember to mention the name of the person to whom you are addressing your question to make it easier for the chair to manage the session at the end. Finally, the last thing from my part is obviously to present today's chair. Ramon Eritja is a research professor at the Institute for Advanced Chemistry of Catalonia, the IQAC, of the Spanish National Research Council, CSIC, located in Barcelona in Spain. He's also a staff member at the Spanish Nanomedicine Network, the CIBER-BBN, and he's the group leader of the Nucleic Acid Chemistry group at the IQAC and the research director of non-biosis oligonucleotide synthesis platform, the U29. He's really interested in the development of a methodology for the synthesis of modified oligonucleotides for therapeutic and diagnostic uses, as well as the study of the properties of non-canonical nucleic acid structures such as G, quadruplex, triplex, and I-motif. Recently, he became interested in the development of DNA biosensors for viral RNA detection and the functionalization of DNA origami to enhance their biomedical properties. Thank you, Ramon, for chairing today's webinar. The microphone is yours. Everyone, enjoy.
Good morning, or good afternoon, or good night, depending on the side of the world that you are. Welcome and Happy New Year, because we are just very close to the change of the year. I would like to, first of all, thank to MDPI and especially the journal Molecules for the organization of this webinar, and especially Heidi Liu and Anna Sanchez. When I was proposed to give this seminar, we thought that it would be a very nice idea to dedicate this nucleic acid chemistry seminar to the memory of Enrique Pedroso. Enrique Pedroso just died in September of 2020, and he is really one of the pioneers or researchers of nucleic acid in Spain. So we do, Professor Pedroso, it's really very make very important contributions in different aspects of nucleic acids, especially synthesis of oligonucleotide conjugates and cyclic oligonucleotides, I just put here just a small mixture of what I believe are the most important contributions, although this is a personal view and somebody could change this selection. I will say one of the nicest results was the development of a growth and support that was able to make cyclizations on the support that opened the possibility to produce large amounts of cyclic oligonucleotides, especially shorter oligonucleotides. And with this technology, he was able to really open a new field, that is the field of the V loops and all these fancy structural species that can be observed only in these cyclic short oligonucleotides. Also, a very important contribution from Enrique Pedroso was the synthesis of oligonucleotide peptide conjugates, and especially the development of protocols for the stepwise synthesis of these compounds. And because his background in peptide chemistry allowed him to tune all the protein groups that are needed for the amino acids to make it compatible with the oligonucleotide synthesis protocols. Okay, so Enrique was also, in addition of an excellent scientist, he was also a fantastic organic chemistry teacher. And of course, he took all the responsibilities, and he was director of the department, and I believe he was also dean of the Faculty of Chemistry for a certain time. But we remember him because also for the participation of all the international societies. Importantly, the international roundtables. He was there all the time, and it was in one of these meetings that was in San Diego in 1996. We got together all the Spanish researchers in this field and decided to make a special biannual meeting also in Spain, and that is what that become what we call RAN. That is the Spanish nucleotides and nucleic acid meetings. I found one of these. The first one was organized for Enrique and Anna, his wife Anna, and this is one of the pictures that I got in Sitges. It was done. Is a town that is 40 kilometers from Barcelona, and was in 1997. And the other picture is just the visit of Manoharan to Barcelona. Is the latest picture that I have, and you see Mano, myself, Enrique, Anna, and Carlos Ciutat. Okay, so we miss him a lot, and we hope rest in peace. And the seminar, I will just would like to introduce what is the importance of the nucleic acids in science. And see, it has been from very beginning, I would say, from the last 100 years, practically, a science that provide the knowledge generated a large number of biomedical applications. And it was a thing that happened from the first isolation and synthesis of nucleotides that generate a large number of antivirals and antibiotics until just the last discoveries that are in the therapeutic field, in where oligonucleotides are able to cure hereditary diseases by exon skipping. That is one of the dreams of I would say, of mankind, that try to solve the problems from people who already have defects in birth. No? And the latest, of course, with the pandemics, we saw the development of the mRNA vaccines. That was an unexpected but fantastic solution for this pandemic. But also at the same time, we have seen one siRNA, a medicine that will be able to be used, because it has been approved in December of last year for the treatment of the high cholesterol, and that is a disease that affects around 5% of the population. So that is really a big moment for nucleic acid chemistry. And for this, I would say you will see that most of the seminar will be dedicated to the search of therapeutic uses of oligonucleotides and nucleic acid derivatives. So that is the speakers that we have here. Today is Daniela Montesarchio, Montserrat Terrazas, and myself. Daniela, that will be the next speaker, is a fantastic scientist from the University Federico II de Napoli, and he has been in this field for quite a few years, and his research is centered also in G-quadruplex and preparation of conjugates, but also he also works with metallic with these metal ions as an anti-cancer products. Before I give the word to Daniela, I was just only to make this small announcement that we have a special issue that is linked to this conference. That is also in memory to Professor Enrique Pedroso, and you can and the deadline of this special issue is 30th of May. But you can apply if you are interested in sending a manuscript in this special issue. And so I will just give the word to Daniela Montesarchio. He will talk us with chemically modified G-quadruplex forming aptamers in biomedical research. You can start whatever you like.
Okay, so thank you, Ramon, for the kind introduction, and good afternoon to everybody. Can you hear me, first of all? And it's really a privilege for me to participate to this webinar in memory and honor of Professor Enrique Pedroso from University of Barcelona. Enrique has been a fantastic researcher, worldwide recognized in the chemistry of nucleic acids. But mainly we will remember him as a wonderful person who unfortunately left us too early. Here there is also a nice picture I found in my records of Enrique with Professor Ana Grandas, his wife and companion, as well as a colleague for life, and other colleagues, Carlos Gonzalez, Aldo Galeone, Tina Giancola, Antonio Randazzo, in a fantastic conference we attended together in Bern, one of the international roundtables on nucleoside, nucleotides, and nucleic acids that Enrique attended and made very, very rich with his contribution. Let's go to chemistry. And of course, I won't have the time and this contribution to discuss about the multiple roles that G-quadruplex structures, this non-canonical DNA and RNA structures can have in vivo. I will mainly discuss about synthetic oligonucleotides able to form G-quadruplex structures, which can act as aptamers, recognizing specific proteins. As you know, the term aptamer is very, very general and refers to any molecule, any compound, which is able to recognize with high affinity and specificity a given target. short RNA and DNA molecules can indeed have these features by folding into very peculiar 3D architectures. And the interesting thing is that aptamers typically recognize proteins and are evolved to recognize in a specific manner proteins, but they can be evolved also to recognize ions, so small molecules, so cells, and even entire organisms as bacteria and viruses. Typically, aptamers are obtained using an in vitro selection procedure, which is named CILEX, which is an acronym standing for Systematic Evolution of Ligands by Exponential Enrichment, which is essentially based on affinity chromatography. Typically, aptamers are considered to be a very promising alternative to antibodies because they offer, in comparison to antibodies, also smaller size, lower immunogenicity, higher stability in a wide range of pH and temperature, and this is a great advantage. They can be much easier chemically modified, so to obtain an increased stability and affinity. The interesting thing is that the literature is full with a lot of aptamers, which have been discovered in the last 10 years. And if you look at the known aptamers, it's interesting to consider that many of them are indeed the G-rich oligonucleotides able to fold into G-complex structures. So the question now arises spontaneously, how come that if we start from large, very large combinatorial libraries of random sequences, we finally end up with a high occurrence of G-rich oligonucleotides, which are fished out from CILEX? Well, there are multiple answers to this question. First of all, most aptamers are evolved to recognize proteins, and many proteins have indeed positively charged binding sites. If we consider this, then G4 structures are indeed optimal for this recognition because they have twice negative charge per repetitive structural unit compared to duplex-based systems. Then in addition, we have to consider that G4 structures are much more polymorphic than duplexes. They are more compact and they also offer a much higher plasticity. This means that they can indeed adapt much easier into a large variety of protein binding sites. On this basis, we have to consider that for in vivo use of aptamers, we typically have to modify them in order to increase their resistance to nucleases, to increase their ability to cross cell membranes, and also to achieve a higher affinity to the target. In the latter case, we have to consider that in most cases, generally researchers act using a trial and error procedure because in most cases we do not know which is the effectively bioactive conformity. I will discuss in this talk essentially about two well-known case studies, discussing about the so-called TBA, thrombin binding aptamer, which recognizes thrombin, and the so-called AS1411, which is an anti-cancer agent recognizing nucleolin. Let's start from TBA. You all know that thrombin is a serine protease playing multiple roles in the coagulation cascade. Particularly, it catalyzes the last step of coagulation, which is the conversion of soluble fibrinogen into insoluble fibrin. And the so-called thrombin binding aptamer, TBA15, also called HD1, is a 50-mer which was identified through CILEX in 1992. Its solution, its structure in solution was indeed solved 1 year later, and it is an aptamer able to fold into an antiparallel chair-like G-quark structure, recognizing exosite 1 of thrombin. When complex to the protein, thrombin is not able to catalyze conversion of fibrinogen into fibrin, and therefore an anticoagulant activity is observed. Indeed, TBA was indeed the first aptamer which entered phase 1 clinical trials as an anticoagulant in vivo on humans, and the anticoagulation effects were observed, but very high doses were required. Therefore, finally, phase 1 studies were discontinued due to the aptamer suboptimal dosing profile. It is clear that the main limit of this aptamer was its poor resistance to enzymatic degradation in vivo. Therefore, many chemical modifications have been inserted into this oligonucleotide so to overcome this drawback. Particular modifications at the level of the nucleobase, of the sugar, of the phosphodiester linkages have been inserted into this compound, but also it has been conjugated, for example, to polyethylene glycol, lipids, and nanoparticles. These strategies indeed have been proved to be really successful. For example, Macogen, which is an anti-VEGF RNA-based aptamer, has proved to be very effective in the treatment of age-related macular degeneration and was approved by FDA in 2004. And as you can see in the structure of this molecule, there is a polyethylene glycol appendage of R' end, a thymidine which has been inserted at 3' end through and 3' phosphodiester linkage, and also the other nucleosides have been modified. So the general modification post-Silex approach is really effective. When we talk about the TBA, it is really interesting to consider that the literature is indeed huge of analogs. A huge number of TBA analogs have been described. Many of them proved to be more effective. Some of them proved to be less effective than the unmodified compound. It's really very, very difficult to resume all the different analogs which have been described in the literature in the last 2 decades. If you want to have more details about this topic, you can refer, for example, to this review which we published last year, and is really an exhaustive picture, at least updated to 1 year ago. What I would like to talk to you today is about how we approached the problem of improving selective recognition in this compound. The idea was to go through structural pre-organization of the molecule by synthesizing a cyclic analog of TBA. That is to say, by connecting through a covalent linker the 3' and 5' ends of the G-quadrex forming aptamer. The idea was to obtain 2 major benefits upon cyclization. First of all, the absence of free termini should protect the oligonucleotide from exonucleases. And then, of course, cyclization should impose a structural pre-organization, which in principle should favor G4 formation. The first linker we designed and prepared is this 20 atoms long linker, which is indeed a flexible one containing, as you can see, a triazole moiety in the middle, which is, of course, the result of copper-assisted azide alkyne cycle addition reaction, which is, of course, the reaction that we realized so to connect the ends of a properly modified TBA analog. The synthesis of this compound was carried out in collaboration with the group of François Morvan, Jacques Casser, and Montpellier, starting from an azido modified CPG support. On it, we assembled the TBA sequence, finally introducing also an alkyne containing building block. At the end, we completely deprotected and released in solution the modified analog, having at the 5' end an alkyno group and the 3' end an azido group by addition of copper ions. So we finally obtained the cyclic compounds in very good yield, which was purified by HPLC, characterized by MALDI, and HPLC again. The cyclic molecule was at this point fully characterized by detailed biophysical analysis, including several techniques, and its anticoagulant activity was determined by DLS analysis. In all cases, we analyzed the Cyc TBA compound in comparison with the modified TBA and also of a linear TBA, which is the immediate precursor of CIBER-BBN, into different buffers. A so-called sodium-rich buffer, that is to say PBS, which is a good mimic of the extracellular environment, and potassium-rich buffer, which is a good mimic of intracellular environment. So to have information about the different behavior in these two different media. First of all, we wanted to check that our new analog CIBER-BBN was able to form a G4 structure, and this confirmation was easily obtained by UV analysis, and particularly by recording the thermal difference spectra. As you can see, in both buffers, CIBER-BBN had the typical signature of a G-quadruplex structure with a minimum at 295 nanometers and a maximum at 273 nanometers. Then, of course, we analyzed the overall behavior, conformational behavior of this compound in the two buffers by CD analysis. Circular dichroism spectra gave profiles very similar to the unmodified compound, to the unmodified aptamer. And CD melting experiments gave us information about the thermal stability of the G4 structure of the cyclic compound, again in comparison with the unmodified aptamer. At all the studied concentrations, here you can see that in both buffers, CIBER-BBN proved to be much more stable than linear unmodified TBA, with melting temperatures which were typically 18 degrees higher. Interestingly, the linear precursor TBA4 was less stable than unmodified TBA. So the presence of these DUNC ligands was indeed destabilizing for the G4 structure. The melting curves were compared to the annealing curves, and as you can see, there was a very good overlapping, showing that the heating and cooling processes were essentially reversible. At this point, we analyzed the behavior of this aptamer by GIL electrophoresis under native conditions. And as you can see, CIBER-BBN showed a single band on the GIL with higher mobility compared to a modified TBA, which is probably due to the higher compactness of this aptamer. And interestingly, even going up with the concentration, there was no evidence of aggregate formation for this aptamer. At this point, we went on with studying the resistance to nucleases of this new aptamer, and we incubated it with fetal bovine serum in parallel with the modified TBA. So here you can see that if a modified TBA was essentially almost completely digested in 2 hours, under the same conditions, cyclic TBA was indeed still present after 7 days. By analyzing the HPL traces, it was possible to determine the half-life times of the new aptamer, which proved to be 180 times more stable in serum than unmodified TBA. With all this data in our hands at this point, we wanted to check the anticoagulant activity of this new TBA analog. And we did that in collaboration with the group of Professor Luigi Paduano in my department, who did a DLS analysis. Indeed, in clot formation, we can easily monitor the formation of insoluble fibrin by following the scattering of light, which is produced indeed upon clot formation. In the presence of an inhibitor, there is a strong reduce in clot formation, of course, and therefore in scattering of light. And therefore, from these curves, and particularly from the slopes of these curves, we can also derive kinetic parameters. So when analyzing the behavior of our new analog, we found out that indeed it showed anticoagulant activity, but reduced by almost half compared to a modified TBA. At this point, we had also confirmation of this behavior by checking the affinity to the protein, and by fluorescence titration analysis, we could monitor the intrinsic fluorescence of the protein by adding increasing amounts of the aptamer. The experimental data were fitted using a one-to-one binding model, and it was possible also to derive binding constants, which indeed showed that the CIBER-BBN had almost half binding constants that a modified TBA. So in conclusion, we had synthesized a very interesting analog, a cyclic analog of TBA, which had a remarkable increased stabilization in its G-quadratic structure, remarkably high resistance to nucleases, but it had reduced anticoagulant activity and binding affinity to the protein. So we reasoned that probably this effect was the result of too much rigidity of our cyclic compound. This is indeed the structure of the first linker we devised, containing 20 atoms from this phosphorus to this phosphorus atom. And we thought of extending this link by synthesizing new analogs, new optimized analogs, having higher lengths. So we synthesized this set of cyclic derivatives. We named CIBER-BBN1, CIBER-BBN2, 3, and 4. The first two obtained through oxime ligation method, and the latter ones obtained again through copper-assisted azide alkyne cycloaddition protocols. Then 4 new analogs have been analyzed again in detail by several biophysical techniques. CD analysis showed that for both of them, conformational behavior similar to the one of a modified TBA, but associated with typically much higher stability in the G4 structures, particularly CIBER-BBN1 and 2 proved to be in both buffers to be much more stable than a modified TBA, with melting temperature typically even 17 degrees higher than a modified TBA. These analogs have been then subjected to a full characterization concerning the resistance to nucleases. So you can see the half-life times proved to be also in this case is much higher than a modified TBA, with the CIBER-BBN1 and CIBER-BBN2 to be the most resistant compounds in the series. At this point, the interesting thing was to check their anticoagulant efficacy, and all the 4 compounds proved to have anticoagulant activity, but only one, CIBER-BBN2, proved to be much more effective than a modified TBA, again by DLS analysis. So this candidate proved to be the best in the investigated series because not only it had increased stability in its G4 structure and a much higher resistance to nucleases, but it also showed improved inhibition of thrombin activity. In this field, actually, we think we obtained a very good compromise between flexibility of this linker and increased structural pre-organization of the G4 structure in this TBA analog. In parallel, more recently, we have also analyzed an additional topic. Again, we have designed and synthesized some new TBA analogs, which have been modified at the ends with aromatic moieties, particularly dialkoxynaphthalene and naphthalene diamides. The idea is to obtain not cyclic analogs of TBA, but pseudocyclic molecules in which the aromatic moiety can stack on top of the other, so that the final G4 structure can be stabilized by pi-pi or charge transfer interactions. So again, in collaboration with the group of François in Montpellier, we designed and synthesized a series of new analogs containing either one set or two sets of aromatic compounds at the 5' and 3' ends. Of this series, the best compound proved to be the one having two sets of NDI and dealkoxynaphthalene residues. This compound, in fact, showed a higher anticoagulant activity compared to unmodified TBA, associated to a much increased stability of its G4 and a higher resistance to nucleases. We are now at a level in which we have two molecules, a cyclic and a pseudocyclic derivative of TBA, which seem to be very, very promising for advanced study. And in collaboration with the group of Menasica, we managed to obtain crystal structures of the complexes with thrombin, and Professor Menasica and her group are now analyzing their crystals to, of course, disclose all the intimate details of the recognition between the aptamer and the protein. So I hope I have new data to show to you in a very short time. I will very shortly now discuss the last topic of my talk. Again, we, of course, aimed at improving selective recognition of aptamers, also favoring the active targeting activity of a very interesting aptamer, which is AS1411. AS1411 is a G-rich 26-mer, which was discovered by the group of Paula Bates and John Trent in Louisville, and also entered phase 1 and phase 2 clinical trials as anti-cancer agent. It recognizes in a specific manner nucleolin, and through this recognition is able to reach nucleus. It's a particularly polymorphic aptamer. In fact, it is a seminal work by John Trent and collaboration with Andrew Lane. The authors were able to separate and identify several different conformations of this aptamer, also including dimeric species. The bioactive conformation is absolutely unknown, but what is clear is that this is an absolutely bioactive compound with anti-cancer properties. Also, some derivatives of this sequence have been studied and found out to be endowed with a high activity. Interestingly, for example, Antoine Fan recently identified a very similar sequence to AS1411, here named Agro 111, 100, with the two T's at the ends, which maintains the same anti-proliferative activity of AS1411, but falls into a unique conformation. The interesting thing is that AS1411 is really effective, no matter what is bioactive conformation. So we decided to study some derivatives, some lipidated derivatives of AS1411, carrying either a stearic acid tail or cholesterol tail attached through a lipophilic or polar linkers. This has been analyzed by a variety of techniques. And the interesting thing is that the lipidated analogs indeed maintain the same overall conformational behavior as AS1411. Also, in addition, they have some reduced polymorphism. As you can see here, in AS1411, there is a band which is attributable to anti-parallel G4 structure, which is not present in this lipidic derivative. The systems have been analyzed by CD melting analysis. Typically, the lipidated derivatives of AS1411 are of similar stability or slightly reduced stability. In many cases, it's interesting to note that multiple processes are going on, probably as the result of the fact that these compounds, upon increasing the concentration, tends to form micelles, as evidenced, for example, by native gel electrophoresis. Here you can see that there are retarded bands of the oligonucleotide, which are due to aggregates with very high molecular weight. This has been confirmed by size exclusion chromatography. And here you can see that upon increasing the concentration, as expected, increases the amount of micelles, and by also the LS analysis. The interesting thing is that when we checked the anti-proliferative activity of these analogs, indeed they resulted to be bioactive against cancer cells, even though their cytotoxicity were a little bit reduced compared to the unmodified aptamers. In conclusion, the lipidase, of course, favor the formation of aggregates in aqueous solution, but the G4 structure maintain their stability. And these lipidated aptamers are a little bit less cytotoxic, but can still be recognized by nucleolin. The idea was to use at this point this lipidated aptamer to carry other drugs, and very recently this approach has been showed also by the group of Carla Cruz, who actually studied the mixture containing this lipidated S1411 derivative of this acridinium binder. The drug is nicely released in cancer cells, and it proved to be only active against cancer cells. So the field is very promising. I would just want to close by citing very, very rapidly nice work in collaboration with the group of Professor Ramon Eritja, in which we proved indeed the ability of this aptamer to act as an active targeting agent. So we prepared niasomes incorporating this ruthenium complex. When in the presence of S1411, the formulations which we studied in their, of course, morphology and stability over time by different techniques, finally proved to be much more effective on cancer cells than on normal cells. And the really interesting thing is the role of this aptamer to specifically deliver the nanoformulation to the cancer cells. So I just conclude now. I hope I convinced you that the G-covered structures are the versatile structural motifs to build aptamers, and that when we identify G4 forming aptamer, generally optimization is necessary, but this is a trial and error strategy, and therefore a lot of work is necessary. However, chemists know a lot of strategies for this optimization. So new solutions have been found. I'll now thank you for your attention, and I want to thank people in my group who collaborated with this research, Professor Domenica Musumeci, Dr. Claudia Riccardi, Dr. Chiara Platella, Dr. Ettore Napolitano, and all the students and all the colleagues in Italy and around the world, with whom I really had the pleasure and the real possibility to collaborate. I now ask you for questions, and again, thank you for your patience.
Okay, thank you very much, Daniela. It's a great talk, as always. I like very much this link G4 because it really will have a very nice opportunity for good activity. Okay, let me just pass to the next speaker. The next speaker will be Montserrat Terrazas. Montserrat did the PhD thesis in the University of Barcelona in 2001, directed by Xavier Ariza and Jaume Vilarrasa. Then he started and make two postdocs, one in University of Leuven with Piet Hedwin, and the second with Eric Poole. And finally, he just returned to Barcelona and had the immense pleasure to have her in my lab for a few years. Then she moved to work with Professor Modesto Orozco, and now just last year he got a permanent position as an associate professor at the University of Barcelona, and now she has his independent group, and I congratulate for that, I must say, because you well deserve that position. Okay, so now he's working in the development of multifunctional oligonucleotides to the treatment of multiple targets. So anytime you can start to talk.
Okay, thanks, Ramon, for your kind introduction. It's really a pleasure for me to be here today to give this seminar in memory of Professor Enrique Pedroso, and this talk is dedicated to him. Well, good afternoon, everyone. As you can see from the title, this talk is going to be focused on the development of an oligonucleotide with therapeutic potential. Well, in fact, as most of you know, over the past 40 years, the use of oligonucleotides to inhibit gene expression and thus to avoid the synthesis of undesired proteins has been the focus of many researchers. And here in this slide, I'm showing you 4 of the most relevant therapeutic oligonucleotides that have been developed, which are the sgRNAs, the short interfering RNAs, also known as siRNAs, the antisense oligonucleotides, known as ASOs, and the triplex-forming oligonucleotides, the TFOs, which activate the CRISPR-Cas9, the RNA interference, the antisense, and the anti-gene silencing and gene editing mechanisms, respectively. And the work I'm going to talk to you today is going to be focused on the development of oligonucleotide tools based on siRNAs, which in order to apply them in the future, so once we obtain the optimal design, to apply them to the other classes of oligonucleotides. So let's talk about RNA interference. RNA interference is a very powerful technique that allows the inhibition of expression of virtually any gene in cell culture. And as I said before, this mechanism is activated by sgRNAs, which are 19 base pair double-stranded RNAs with dinucleotide overhangs at the 3' ends that are formed by a sense and by a guide strand that is complementary in sequence to a fragment of the target mRNA. These molecules can be generated inside the cells by dicer cleavage of longer double-stranded RNAs, or alternatively, we can synthesize them in the laboratory and incorporate them into the cells by using cationic liposomes, such as lipofectamine, which can be used in cell culture, but not in vivo due to their toxicity. And once inside the cells, they are recognized by a multiprotein complex that is called RISC, which unwinds the duplex and loads the guide strand into the complex and uses it as a template to find the complementary target mRNA, and then it induces its cleavage, preventing thus its translation into protein. Thus, as you can see, all these properties make sgRNAs very good candidates for the treatment of many diseases, including cancer. The problem is that the clinical application, and in fact of all the other oligonucleotides in general, faces important limitations. One of them is their low ability to cross the cell membrane due to the negative charge. Another one is their poor biostability due to their susceptibility to degradation by serum nucleases such as C'-exonucleases, endonucleases, and RNases. And in the case of cancer therapies, there is still another limitation that needs to be taken into account, which is the appearance of drug resistance, which is characterized by the activation of a secondary pathway of the tumor, here in red, in response to the blockage of the major pathway in green, due to the action of a drug in such a way that the activity of the inhibited pathway is recovered. leading to the proliferation of the tumor. Well, in order to overcome one of these limitations, the problem of the low biostability of oligonucleotides, many chemical modifications have been developed in the literature, such as 2'-fluoro, 2'-O-methyl, 2'-O-methoxyacyl substitutions, as well as phosphorothioate bonds. And these modifications can be easily incorporated into the oligonucleotide by using an oligonucleotide synthesizer and by applying the phosphoramidite cycle, which starts by removal of a dimethoxytrityl group at the 5' hydroxyl group of a nucleoside attached to a solid support. Then, the addition of an activated nucleoside in the form of a phosphoramidite leads to the formation of a phosphite triester bond. And the unreacted nucleosides on the solid support are capped by the derivatization of the 5' hydroxyl group to the corresponding acetyl ester. And finally, the phosphite triester bond is oxidized to the phosphotriester bond. And multiple cycles of this approach leads to the formation of the oligonucleotide of the desired length and sequence. On the other hand, in order to improve the cell uptake of oligonucleotides, many strategies have been explored. For example, the conjugation of lipids or positively charged peptides to the oligonucleotide, as well as their encapsulation into lipid nanoparticles. In fact, this approach is very promising and is the one that is currently used to administer one of the sgRNAs that have been approved for in vivo use. The problem here is that due to the lipidic nature of these nanoparticles and to the fact that they are not functionalized with ligands targeting specific receptors, they accumulate in the liver. And that's why most of the oligonucleotides that have been approved, as you will see in the next slide, are targeting genes that express in the liver. As an example, here I'm showing you the three sgRNAs that have been approved for in vivo use, and the 7 ASOs that have also been approved. As you can see, all of them are modified with this biostable modification that I showed you before. And in the case of the sgRNAs, for example, they are administered via either encapsulation inside lipid nanoparticles or by conjugation to a ligand that is targeting a receptor expressed in hepatocytes. Thus, as you can see, much work has been done in this field, but still much work still needs to be done in order to apply these oligonucleotides for the treatment of cancer. In fact, ideally, in order an anti-cancer therapy based on oligonucleotides should meet the following requirements. To be able to selectively deliver the oligonucleotide, which should be biostable, to the tumor cell, leaving intact the healthy cells, and to be able to overcome drug resistance. And these have been the goals of our research for the last 10 years. And that's what I'm going to talk to you today. Well, but to develop these tools aimed against the cancer, it is clear that we need to address all these limitations one by one. So let's start with the first one, the difficulty to cross the cell membrane. Well, a promising approach to improve the cell uptake of sgRNAs could consist in the covalent linkage of the sgRNA to a ligand, such as a peptide targeting a specific receptor in order to, express in a tumor cell in order to induce the selective delivery of the sgRNA to inside the tumor. However, despite the great therapeutic potential of this approach, the problem is that the synthesis of these sgRNA peptide conjugates can be problematic due to problems of functional group compatibility during the solid phase stepwise synthesis of these conjugates. But an alternative approach could consist in synthesizing separately RNA and peptide fragments functionalize with a potentially reactive group and then chemoselectively react them in solution. And among the different strategies that have been developed, we all know that the azide alkyne hexane cycle addition is very efficient and selective. In fact, this strategy has produced a large number of DNA peptide conjugates. But the problem is that due to the liability of RNA, due to the presence of 2' hydroxyl group, limited research has been done in this area. In fact, when we started to work on this project, only 2 sRNA peptide conjugates had been synthesized, and they contain very small peptides. Well, but in collaboration with Dr. Vicente Marchal from the Organic Chemistry section at the University of Barcelona, we developed a new approach for the clinical functionalization of sRNAs with very large and functionalized and complex peptides recognized by receptors expressed in major tumors. In particular, we focus on the functionalization of the sense strand of an sRNA targeting the HER2 gene with 3 different peptides. With the cyclopentapeptide RGD-FK, which is recognized by integrin alpha V beta 3, that is expressed in many tumors, among them the SK-MEL28 melanoma cells, which also express acceptable levels of HER2. The other peptide was octreotide, which is a cyclooctapeptide that contain disulfide bridges and trifunctionalized amino acids, and that is recognized by the somatostatin receptor, SSTR2, which is expressed in many tumor cells, among them the SKB-R3 cell that express the HER2 receptor. And the other peptide was this one that they called CPAHNP, which is a chimeric peptide composed of a self-penetrating domain and an AHNP domain that is recognized by the HER2 receptor. Our approach consisted in the reaction of this alkylated sense strand targeting the HER2 mRNA with the azido derivative of the corresponding peptide in the presence of copper sulfate, sodium ascorbate in triacylonitrile. And by applying this approach, we observed the complete conversion of the alkylated sense strand to the corresponding sense peptide conjugates. As you can see in this gel electrophoresis analysis, in this first lane corresponds to the alkylated sense strand, and this other one to the reaction mixture. As you can see, in all cases, the major product is the desired conjugate. And then, after HPLC purification and hybridization of this sense peptide conjugate to the guide strand, the resulting sRNA peptide conjugates were able to enter the selected cell lines expressing the corresponding target receptors in the absence of transfecting agent and inhibit the expression of the target gene, which is HER2. For example, here in this case, when we treat the SKB-R3 cell line, which express the HER2 receptor, well, the HER2 gene and the SSTR2 receptor, we observe with this octreotide sRNA peptide conjugate, we observe a significant decrease in the expression of HER2. And what is very important is that this method is very selective because, as you can see in this plot, when we treat a non-tumor cell line with sRNA linked with CPAHNP peptide, which targets the HER2 receptor, we do not observe a decrease of expression of renila, which is the gene that is targeting this sRNA, due to the lack of receptor in this cell line, which confirms the selectivity of our approach. So in summary, as a summary of this first part of the talk, with this work, we had developed a new method for the functionalization of sRNAs with peptides targeting specific receptors expressed in tumors, and to induce the selective delivery of these conjugates to the tumor cell. So in view of these good results, we moved to the second part, well, to our second aim, which was to improve the biostability of the oligonucleotides in order to develop our new tools for the treatment of cancer. Well, in the literature, most of the approaches that have been described to increase the biostability of sgRNAs involve incorporation of a large number of 2' fluoro and 2' methyl substitutions along the sgRNA. And this can lead to a decrease in activity due to contact of these modifications with the risk protein complex. To overcome this limitation, what we did was to design a chemical modification that with minimal perturbation, that induce minimal perturbation of the sgRNA and only protected the ends from nuclease degradation, in particular from 3' exonuclease degradation, which is the major nuclease activity in serum. This modification consisted in the replacement of the dinucleotide overhangs of the sgRNA, in which the two terminal nucleosides are connected by a phosphodiester, by a dimeric nucleoside, which I called VCN, in which the two nucleobases are connected by an alkyl chain, where N is the number of atoms of the alkyl chain. In fact, when we perform molecular dynamic simulations of this dimer, in particular a VC2 dimer connected to an RNA strand by a phosphodiester inside the 3' exonuclease active site of the Krenov fragment, the simulations suggested that we could be moving in the right direction because we observed how this dimer disrupted the binding site by state clashes between the alkyl chain of the dimer and leucine 361, forcing the phosphodiester bond that connects the dimer to the RNA to move far away from the metal cations that are responsible for 3' exonuclease activity, suggesting an inhibition of hydrolysis. Thus, the next thing we did was to synthesize this dimer by protecting the hydroxyl groups of thymidine by a TBDMS group, then by activating the C4 position with a triazolyl group, and by treating the resulting O4 triazolyl derivative with an excess of ethylene diamine. And then the resulting aminonucleoside was reacted with an O4 triazolyl derivative of the thymidine with the 5' hydroxyl group protected by a DMT group and the 3' hydroxyl group protected by an acetyl group, which was removed afterwards to give the dimeric nucleoside with the 5' hydroxyl group protected for RNA synthesis and the 3' hydroxyl group available after removal of the acetyl group for conjugation with the solid support. Then we used this dimer to incorporate it into the 3' ends of each of the strands of an sgRNA targeting the BCL2 oncogene. And stability studies in the presence of a 3' exonuclease, the snake venom phosphodiesterase, confirm our prediction, since as you can see in this 2-real electrophoresis, our sgRNA is more stable than an sgRNA containing a phosphorothioate bond at the 3' end. As you can see, after 72 hours of incubation, our sgRNA remains intact while the other one is partially degraded. Moreover, our BC2 modification was accepted by the RNA interference machinery, since the Western blot analysis showed activity, or inhibitory activity of BCL2 comparable to that obtained for the natural sgRNA. Thus, in view of these good results, what we did was to explore these dimers in more detail. In particular, we perform molecular dynamic simulations of an sgRNA containing these dimers at the 3' end. And interestingly, when starting from an intermolecular stack conformation, the trajectories show how the dimer evolved to a completely extended conformation, resembling a base pair, which suggested us that we could use these dimers to connect the ends of the double-stranded RNA in order to form a dumbbell-shaped RNA with the ends connected with these caps. In fact, when we simulated these dumbbells, the trajectories revealed that these caps do not alter the global geometry of the double helix, and that the optimal length of the alkyl chain of these BC caps was an equivalent of 6 carbon atoms. Thus, the next thing we did was to synthesize these dumbbells. To do this, we prepared the corresponding dimeric nucleoside, now with the hydroxyl group protected, to incorporate them in the in the loop position of two RNA hairpins with 5' terminal overhangs with a phosphate group, which we linked covalently by enzymatic ligation, which took place with high efficiency, as we can see here. And finally, stability studies in cytoplasmatic cell extracts confirmed also our predictions, since, as you can see in this gel electrophoresis analysis, our BC6 dumbbell is significantly more biostable than a dumbbell described in the literature that contains terminal loops composed of natural nucleosides linked by phosphodiester bond. Moreover, these BC6 dumbbells were found to be good substrates for the enzyme dicer, which clipped this RNA prodrug very efficiently to give the active 19 base paired sgRNAs, which could be used to inhibit the expression of a relevant oncogene in HER2-positive breast cancer, which is GRB7, with longer and prolonged activity than the dumbbell that had been described in the literature, the one I showed to you before. As you can see, after 6 days of incubation of HER2-positive breast cancer cells with this dumbbell, the inhibitory, well, we can see a significant inhibitory activity of GRB7, while the activity of the dumbbell described in the literature was almost lost. So as a summary of this second part of the talk, these dimeric nucleosides allowed us to, which were designed rationally, allowed us to create new nanostructures with high biostability and with high activity. Well, then with these good results in hand, what we did was to move to the third aim of our work, which was to develop new strategies in order to try to help to overcome drug resistance in cancer. Well, a promising approach to do this could consist in the simultaneous blockage of the major and the rescue pathway of the tumor by using two dumbbells. But when we administer a one-to-one ratio mixture of RNAs, we cannot be sure that the same amount of each of them enters the cell due to different transfection efficiencies. But our proposed strategy consisted in connecting the two dumbbells in the same molecule, which at the same time could have additional properties in order to improve the transport of the resulting nanostructure as well as its detection, among other things. And with this idea in mind, what we did was to design this branch RNA, which we call 2shRNA, which is composed of a central part that connects two arms, which are two short hairpin RNAs of different sequence targeting each of them one of the pathways of the tumor. By using this approach, we should be able to administer the two RNA drugs simultaneously. As you can see, each of the parts of the branch RNA is functionalized with a BC6 dimer, which at the same time contains an alkynyl group for click conjugation by using the approach that we developed before, two accessory molecules to give to the final nanostructure the additional properties that I mentioned before. Here I'm showing you a detail of the composition of this branch RNA. It is composed of two short hairpin RNAs that have sticky ends that form base pairs with the sticky ends of the components of the central part. And here I'm showing you how we incorporated the alkynyl group into the dimer by incorporation of a formyl group and treatment with the Westman-O'Hara reagent. And each of the components of the branch RNA can be functionalized previously to the formation of the branch structure with an accessory molecule, which can be, for example, a peptide recognized by a tumor receptor to induce the selective delivery to the tumor, a fluorophore to monitor its transport, or with another accessory molecule, for example, a drug. Then, after functionalization of the components of the branch RNA, the hybridization of the three of them gives rise to the formation of the branch structure with high efficiency. As we can see in this native gel electrophoresis analysis, the first four lanes correspond to each of the components, the building blocks of the structure, and the last lane corresponds to the branch RNA, which migrates more slowly than each of the building blocks and has the same mobility than a branch RNA that we synthesize by covalent linkage of the three components. The resulting multifunctionalized nanostructure was able to enter the cells, the tumor cells selectively, and then once inside the cells, it is recognized by DICER, which cleaves it in order to release the active 19 base pair double-stranded RNAs. Well, and we use this approach to synthesize a branch RNA targeting two relevant oncogenes in HER2-positive breast cancer, which are HSP27 and GRB7. These two genes are overexpressed in HER2-positive breast cancer cells that are resistant to the drug lapatinib. And it has been found in the literature that when we inhibit the expression of these two genes, there is a decrease in the viability of HER2-positive breast cancer cells and an increase in the susceptibility of drug-resistant cell lines, such as this one, the one resistant to lapatinib. There is a susceptibility to these drugs. So, yeah, and as a first approximation, what we did was to synthesize a naked 2S RNA that is without accessory molecules attached to its structure. Here in this plot, the plot of the left represents the cell viability profile of a lapatinib-resistant HER2-positive breast cancer cell line, the UACC732. The blue bar represents untreated cells. In the dashed bars, cells treated with the drug lapatinib. As you can see, the cells remain almost intact in the presence of this drug due to their acquired resistance. When we treat the cells with a mixture of sgRNAs targeting our target genes, a 1-to-1 mixture, we only observe a small decrease in cell proliferation. And this decrease is a bit more pronounced when we combine the mixture of sgRNAs with lapatinib. But the proliferation of the cells decreases dramatically when we treat them with our branch RNA. And as you can see here, lapatinib, which is not active in the cell line, recovers its activity when it's combined with the branch RNA. And what is very important is that these structures are not toxic in non-tumor cells. As you can see in this plot, which represents the cell viability profile of this non-tumor cell. So in view of this good results, what we did was to proceed to functionalize these branch structures. Well, here, what we did was in order to confirm the co-delivery efficiency of this structure, what we did was to attach a CY5 dye of red emission to one arm of the structure and a FITC dye of green emission to the other arm. And then we treated HER2-positive breast cancer cells with this construct in the presence of lipofectamine. As a control, we prepared a 1-to-1 mixture of sgRNAs functionalized with the same dyes. And again, we treated the cells with this mixture. Blue represents nuclei staining with H. As you can see in the first panel, when we administer this branch RNA, our branch RNA to the cells, both dyes co-localize in a single point, giving yellow emission. In contrast, when we treat the cells with a 1-to-1 mixture of sgRNAs, we observe a random distribution, and the intensity, the fluorescence intensity is lower than in the first case. This results confirms that our branch construct is able to enter the cell as a whole construct and administer the two drugs, the two arms simultaneously. While in the other case, it is very difficult to control the concentration of the amount of sgRNA that goes inside the cell. Thus, these results are confirming the robustness of our approach and its advantages over conventional sgRNA strategies that are based on the administration of 1-to-1 mixtures of sgRNAs. Then the next thing we did was to incorporate the CP-AHNP peptide to the central part of the branch RNA containing these fluorescent dyes. As we can see, what we did was to treat HER2-positive breast cancer cell lines with this construct in the absence of lipofectamine. As you can see from the green and red fluorescence emissions, the whole construct entered the cell in the absence of transfecting agent with high efficiency. And when we do the same, in the absence of lipofectamine, but with an analog that does not contain the peptide attached to the structure, we do not observe a fluorescence intensity in the cells, which means that the fluorescence that we observed before was due to receptor-mediated endocytosis, which confirms the selectivity of our approach. And once inside the cells, this branch construct, functionalized with the targeting peptide, was able to inhibit the expression of the two target genes, leading to a significant decrease in cell proliferation of HER2-positive breast cancer cells. Thus, as a summary of the last part of my talk, with this work, we have developed a new tool for the delivery of two RNA drugs at the same time, which can be functionalized with different accessory molecules in order to improve its delivery and in order to monitor its delivery also. And what we are doing now is to apply these strategies to the other classes of therapeutic oligonucleotides. Well, finally, I would like to thank the organic chemistry section at the University of Barcelona the Nucleic Acid Chemistry group at the IQAC CSIC, Molecular Modeling and Bioinformatics at the IRB, and the Instituto Salud Carlos III for funding. And of course, thanks all of you for your attention.
Okay, it's great. I mean, if somebody asked me to make an siRNA, a cyclic siRNA, they say you are crazy. But if somebody asked me to add 2 siRNAs with 3 targeting sites, this is impossible to do. So congratulations for this fantastic work. Okay, and now we have a few questions already. If you like to see, do you want to answer now?
Yeah, I can do it.
Okay, sure. It's a question. You see the Q&A section that says Ana Di Porzio?
Yes.
Yeah. Yes.
So do you think that conjugation with a peptide for peptide receptor recognition could also be exploited to selective delivery of other entities? Yeah, I think so. Yeah. You mean with G4 ligands? Well, it depends on the size. It depends on the size of the ligand, of course, and of the functional groups that it contains. Yeah, but if it contains an alkyl group, it should be able, yes. And the other one is from Daniel Pulido. I want to say thanks for this amazing talk about these visit loops. Do you think that they could be general tools for any sequence, or they need to be optimized for every sgRNA? No. Yeah, that they can be general for any sequence. Yeah, of course. Yeah, because we have applied this modification, so we have incorporated these loops to many other sequences. Yeah, sure. Another one, Luke Ferreira, regarding the approach for cell entry. I imagine that if you have a very strong binder for HER2, the sgRNA could not separate from the protein. Do you have to play with the peptide affinity to release the sgRNA, or would it be possible to add a cleavable linker to release the sgRNA inside the cells? No, really. What we did was to try with the different doses, and well, when we observed that when we increase the dose of the sgRNA conjugate, we observe an increase in fluorescence inside the cells. So we didn't have to play with a cleavable linker.
Okay.
Okay.
So these are all the questions. Thank you very much. Now I will give my talk. Okay, thank you very much. Also, I will. These 2 talks are fantastic. Now I will try to give this presentation in honor to Enrique Pedroso. Yeah, first thing that I will try to do is introduce to the field of the DNA nanotechnology. So, and I cannot start any talk without saying, remembering Ned Seaman, that he is the pioneer of all this. He was a crystallographer that tried to instead of having crystals of proteins with DNA, a specialist in DNA protein complexes, and instead of making crystals of proteins with substrates, DNA substrates, he tried to make lattices, networks of oligonucleotides where the proteins will bind to the DNA. We'll find his specific sequence and I will bind that. This simple thinking make him to open a new field that is the DNA nanostructures, and he did succeed in this field, making all these what is known as a DNA style systems that they can polymerize and make two-dimensional and three-dimensional structures. Yeah? Unfortunately, Ned died just in November of last year, so we just we are very sad for his loss. Then I would like also to introduce one of the most significant discoveries in this field, that was the field of the origami DNA. This was discovered by Paul Rodman in this Nature paper of 2006. He used M13 circular DNA, making with a mixture of 200, 300 oligonucleotides that were designed to fold the DNA in a certain manner, because these oligonucleotides or staples, they have this hairpin stable, and what it does, if they are very well designed, what it does is to turn the M13 DNA to make any figure. So he did hypothesize that he can, with the same M13 DNA, make different shapes by just selecting different groups of oligonucleotides, staple oligonucleotides. This was what he wanted to do, and he designed the oligos, made the oligos, introduced it, and he was able to see that he got whatever he wanted. So he was really, it is behaving properly. So that is what is the advantage of all this is this technique so valuable, is that you add the M13 DNA with an excess of the staples, and these staples are not needed to be purified, because the thermodynamics will make this thing to fold only with the correct oligonucleotides. The incorrect oligonucleotides do not get incorporated. So they are very easy to separate these nanostructures from the staple oligonucleotides. So that was a very fantastic technology. That it allowed to prepare oligo objects, let's say oligonucleotide objects that are around 100 nanometers. That is the size of the M13 duplexes. So then, at the same time, the field become very professional, especially the nanosciences people, to obtain nano objects that are of different contents. Could be from lipids to metal to different polymers, and this could be made with a very homogeneous way. So the field of the material sciences was very successful in obtaining these nanoparticles in exact size. So then they start to think that these nanoparticles could be functionalized with drugs and with targeting ligands, so they can really it will open a new field in the field of therapeutic, that is the nanomedicine. So you could use nanomaterials that are precisely made, exactly functionalized, and to deliver the drugs to the exact tissues that are needed. So these are there's a big and there's a whole special area that is very exciting. So in addition here, for example, we have one lipid nanoparticle that was produced by Santiago Grijalvo. We call it these niosomes that were mentioned by Daniela, and you see that you get a very nice round homogeneous products. This contains a siRNA derivative for the delivery of siRNA oligonucleotides. You see that the dispersion, although it is a nice preparation, you see that there is a certain dispersion, and of course we cannot ensure that all the nanoparticles have a similar content. So that the functionalization of the nano objects and the homogeneity is a main issue in this field. So we for this, when the DNA nanostructures appeared, you know, for example, like the DNA origamis, you know that you can make origamis, and you know exactly the position of these 200, 300 oligonucleotide staples, and you could use that to bring any of the, a combination of drugs or the targeting ligands that will help to the delivery issue. So that is the main issue. And the good thing between these two fields is that the size are similar. So all are in near the 100 nanometers. So although nanoparticles could be made about 40 to 50 nanometers too. So and this is what is the actual research effort. So we have here in this review of 2 years ago, you can classify the DNA structures in as these are the tile systems from Seaman. Also, you could use a small three-dimensional structure like the tetrahedron or the dodecahedron or the blockidal, and of course you can get to a more bigger structure like the origami that are M13 based, and these are 100 nanometers. And with that, you can have different shapes and sizes. Yeah, of course, this competes with all the nanoparticle, the standard nanoparticle field that that uses metallic and non-metallic or lipid or polymer matrices to do this size. So what is important to notice is that the DNA structures and the DNA and the nanoparticles do not function as a normal drug where you have passive diffusion, but they enter to the cells with a very specialized cellular uptake mechanisms. So these are the three that has been known, that are the caviole-mediated endocytosis, the clathrin-mediated endocytosis, and for bigger size nano-objects, the micropinocytosis. So this, with all these precedents, I just selected the two most exciting results in the use of oligonucleotides for drug delivery. And this was a paper of 2012 from Lee and Manoharan. He used the tetrahedron structure to link the siRNA. It's kind of what Montse was trying to do, no? But now, instead of being everything together, it is the assembly of this structure what it puts everything together. They are not chemically linked, but let's say self-assembled structure. This is RNA had this folic acid at the ends that will that directs these structures to the target tumor cells. And this is another, still is a fantastic example of what's called a nanorobot, where it uses the rectangular origami, but this rectangular origami is being engineered to be or tubular or open. And this is controlled by the antisense oligonucleotide that was mentioned by Daniela, the AS1411, that is very sensitive to nuclein that is overexpressed in cancer cells. So this origami holds the drug inside, and when arrived to the tumoral cells, it opens and delivers the drug. This was proved to happen in mice, but also in small pigs. So that is a little bit of well, when you compare the DNA nanostructures and the nanoparticle field for drug delivery, you see that DNA nanostructures could have certain advantages, like defined architectures, reproducible and controllable size, exquisite control of drug loading and receptor-mediated substrates. They are not immunogenic, but they are degradable. So there is no accumulation of toxic material. So in the contrary, they are maybe sometimes too labile, and they can be hydrolyzed before arriving to the tissue and are expensive compounds. So the next thing that I would like to introduce is our drug in these studies. Montserrat has already explained a fantastic approach, let's say one of the most successful therapeutic oligonucleotides that are the siRNA duplexes. These target the messenger RNA, and this makes that an overexpressed protein cannot be produced anymore. But there is different approaches, as Montse explained. You have the antisense, you have the TFOs, and you have the aptamers. All these have a different mechanism of action. And there is a very rich type of oligonucleotides that could be therapeutic. But we use one that is not very usual, so something very strange, but we say very, very important for us. That is what we call the antiproliferative oligomers. This idea came from Gamer that did work with the deca-nucleotide of fluoxuridine. And 5-fluorouracil are well-known antiproliferative compounds, and they do enter by diffusion. And then you have a series of metabolic steps that makes the monophosphates and finally the triphosphates, and these are the compounds that are medicinal because they inhibit the metabolic the formation of thymidine by inhibiting thymidine synthase, and also some of the ribonucleotide synthesis. So, and of course, if it gets incorporated into DNA, they can act as an inhibitor of topoisomerases, and it makes DNA have no sense. All this is known that, unfortunately, with the time, cancer cells learn to pump out all these drugs and to cancel the phosphorylation of the nucleoside. These two are two of the mechanisms that are known to happen in cancer cells when they get resistance to these drugs. So Gemeiner thought that if you add an oligonucleotide, this oligonucleotide, when it enters to the cells, will be degraded, of course. But when it gets degraded, it will form the phosphorylated nucleoside. That is the one that is active. So escaping the pumping out and the inhibition of the phosphorylation that are the drug resistant mechanisms. So, and that is, for us, it was very important to select that, because when you try to work with DNA nanostructures, it will be for us better that everything gets destroyed when it's inside the cells and generate the prodrug. So that for us, it is a good approach for the DNA nanostructures. And that's why we started this work a few years ago, and we select 3 nanostructures, or say nano assemblies. One that is made with 4 oligonucleotides, that is the quadruplex that Daniela explained. This quadruplex is not antiparallel intramolecular, but is a tetramerization of a single oligonucleotide that makes the G quadruplex structure. The tetrahedron that was explained also before, and the DNA origami that was explained before. What we have in all these cases, we have as a drug an oligonucleotide that contains several units of fluoxuridine, let's say 5 in this case and 10 in this case. In addition, there is space to introduce cholesterol to increase the cell permeability of these compounds. And what we have here is that, if you see, have 3, each one of these is 1 order of magnitude bigger. So although they look here the same, these are only 2-3 nanometers, these are 15-25 nanometers, and they are a little bit higher than 100 nanometers. So, and our aim is to study the internalization and antiproliferative properties in selected cell lines. I will start with the parallel G quadruplex. This, as I told you, it is a simple, is the simplest, because one single oligonucleotide, as long as it has a few guanines in a row, it will form this G quadruplex. And then we can continue with a therapeutic oligonucleotide that could be even antisense, or could be FDU oligomers. We have also another position, that is the other terminal end, to introduce the delivery enhancer. It could be peptide or lipid. And the assembly process is like in the others, it's just heating, annealing, heating and let it cool, and it forms this species. So this this assembly is very simple, and but it could have certain disadvantage that we check. That is a potential toxicity that has been described for G quadruplex, because they say that by hydrolysis they will produce a large amount of GMP, and this could be toxic. But I would say that this depends a lot, and in the ones that we use, this is not a problem. And the other important issue here is that in cells, there are a lot of membrane cells, membrane proteins, especially in tumor cells, that contain a large expression of G quadruplex binding proteins. So in principle, the G quadruplex is not only an assembly protocol, but also it generates a compound that is recognized by the membrane receptors of cancer cells. So that is what we did. We just prepare one oligonucleotide, well, say a series of oligonucleotides that contain 5 fluoxuridines, and it could contain 4 or 6 tetrads. And we have, of course, a control sequence where these G's were changed by T, so cannot form quadruplex. So that's for all these studies, we use FDU resistant colon cancer cells. So, and this is, we went to the bibliography. There is several cancer lines, and we select one that is mid resistant and one that is strongly resistant. So these have learned to avoid the nucleoside and the 5-fluorouracil, and for this they do not respond to these drugs. And we wanted to see if we could return this sensitivity to FDU by using these oligomers linked to these nanostructures. So, and this is the first thing is to make the oligos, of course, and then you should characterize, and you see it's very easy to characterize G quadruplex if they are short, because you have a band in the CD that is only present if the G quadruplex is formed. Then you also can do it by gel, by gel electrophoresis in natural conditions, so non-denaturing, and with that you see the bands are a little bit retarded compared to the non-quadruplex forming oligonucleotides. A fancy thing that we observe is that when we stain the gels with the stain gel, even you can see whether it's quadruplex or not by the color. the color of the G quadruplex are much more intense than the non-quadruplex forming. That's something unexpected. Then what we went to see to analyze the internalization of these quadruplexes, and what we do for that, we label the oligonucleotide with fluorescein, and then we go to the cell counter and see how many cells are getting fluorescent because they enter. So, and you see there is a large increase when you get the G4 or the G6 sequence. What if you have a T, you don't have any entry. And if you do the controls, TG4T or TG63, these are without fluoro-U, but you show that is the formation of quadruplex, what it drives the cellular uptake. And you can get up to 80% of the cells labeled after 24 hours. Then we went to analyze the antiproliferative result using what is called the MDPI assay. And you see that after 24 hours, the oligonucleotide that has no FDU are not cytotoxic. So the toxicity assigned to the G and P formation is not great. But when you have the cells in FDU and quadruplex is when you see the antiproliferative activity. These are G4, G6, and this is 10 FDUs instead of 5, and we see that's the best combination. Then we did another experiment that is called the analysis of the cells after 48 hours, whether they are apoptotic, later apoptotic, or necrotic, and that confirms that this cytotoxicity comes from expected from the FDU. Okay, so then we went to the DNA tetrahedron and the DNA origami. For the tetrahedron, we just had these oligonucleotides that we just make just heat it and cool it down, and depending each of these oligonucleotides, ones will have cholesterol and others will have F10 fluoxuridines. And with that, we will make a variety of nanostructure with a different amounts of cholesterol and FDU. For the origami, we do the same approach. We have now the staples, because this is a bigger structure. We can include much more FDU units and much more cholesterol units. Unfortunately, there is also another problem is that these units so many oligonucleotides and the M13 that we cannot work with high concentrations. We use our working concentration is 100 nanomolar, and with the tetrahedron, I have the working is about 5 micromolar. Such a difference. So these are all the potential structures to study with 1 fluoxuridine or 2 fluoxuridine and a different number of cholesterol, 2, 3, and 4 molecules of cholesterol in the tetrahedron side. And in the origami, we have, imagine these 4 deca fluoxuridines, all 6. This will amount 60 molecules of fluoxuridine in the structure with a different number of cholesterol units. Then the first thing is just to check that everything falls correctly, because the addition of these extra things in the staples could prevent the origami formation, but we see that by gel we can see that there is no change on the mobility, and you have this strong band that indicates that the origami has been formed. This is also these things are big enough to be visualized very well by atomic force microscopy, and you see that it's you see all these nanostructures all equals, and each one of them have the same concentration of cholesterol and FDU. With the tetrahedron, we also check by gel. This is an agarose gel. This is a polyacrylamide gel, I believe, and we see the formation of the expected band. We can. This is much more difficult. It is too small to be to see it by FAM, but we were able in one of the samples we were able to see and check that is the right size. As another technology to check the homogeneity of this preparation is the use of zeta sizes, and we see for the tetrahedron you get the average that you expect, the size that you expect, and everything was the same for all except for this one that contains 4 cholesterol. In this 4 cholesterol structure, we start to see a bigger object. That most probably is some aggregation of these tetrahedrons due to the cholesterol content. Looking in the cryo microscopy, we could even observe a dimer showing that this is really aggregation. Okay, so in the origami, there is no aggregation at all. It's just only a small increase of size. So the unmodified origami have a size of about 124, sorry, and the full functionalized with a lot of FLU and a lot of cholesterol, it has a little bit bigger, 141. So the cellular uptake. So we could see that these oligonucleotides enter to the cell, and what we see, that's the tetrahedron, and you see that these are labeled with C13, and we can see it inside of the cell. And the cholesterol, it seems that are much better. The cellular uptake are much better. In the origami, we could not use the C13. We use cyber green staining, and you see that there is a big difference between the origami that is not functionalized. The entry is slow, and there is not much green points. But in the full cholesterol, you see a large cellular uptake. So this is already the activity, the MTT activity. We see with these FDU resistant cells. We see in the tetrahedron the best is this one that contains one decamer of fluoxuridine and 2 cholesterols. It is funny that the ones that have more cholesterol, it does not get so much activity, probably due to this aggregation that we saw. And the ones also with more FDU also seems not to increase the activity. So there's a balance between the amount of drug and the cholesterol containing. And in these are the less resistant cells. These are the highly resistant cells. You see it immediately. These are the controls, are the nucleosides. These are practically not cytotoxic at these concentrations. But when you add the tetrahedron at the same concentration, you see that the one that has 4 cholesterols now, it is much better. So in the case of the origami, in general, we see less activity because the concentration that we could use is much lower. But you see, we can see that the one that contains 40 fluoxuridines and 4 cholesterol is the more active, and in the HSCs, in the more resistant cells, the activity is not so great, but still we see a clear increase in the 40 fluoxuridines and 4 cholesterol. So then with this apoptosis analysis, we see that also this the cytotoxicity of this species, we really see that the origami that was not so clear in the MTT experiments, now with this analysis of the apoptotic cells, it becomes higher, even with being at least less concentration. And this is because of the large amount of drugs that contain this structure. Now, finally, we get this analysis that's called the tumoral cell growth assay. Here you put the samples and the cells, and after 24 hours, you try to make it grow. And of course, if the cells are not correct, they don't grow so well. And after 8 days, you see more or less clones from this. And this is much more sensitive, and you see now that in HTV 38 cells, the number of colonies are very low after all this long treatment, and in the more resistant cells, you also have a nice inhibition. This is, in this case, the best is the one that has 2 cholesterol and 1 decanucleotide, and in the other, the best, the higher of cholesterol, the better. In the origamis, you have the same similar results, but are not so evident than the ones that we observe with the tetrahedron, because we work here in very small amounts. But anyway, we see the highest activity with the 4 and 6 fluoxuridines and high cholesterol content, and with the more resistant cells the same. So finally, just as a conclusion, I think we were clear that that the nanoassemblies functionalized with the FDU are more cytotoxic than the conventional drugs at low concentration, at least in these cells that are FDU resistant. The cholesterol seems to play an important role, increasing the activity, and we believe that that the increase of toxicity it helps to overcome the resistance of the cells. We have a very good cytotoxicity effect, but tetrahedron display the best antiproliferative action, at least compared with the DNA origami. Finally, I would say that for us, all this was a very nice lesson that DNA structures could be a good delivery agent. But still, we believe, because you need to work with these so small amounts, we need a much higher activity to become practical somehow. And for this, our future directions are in two sense. One is the incorporation of several drugs in one of the structures, so that will be combined therapy. For example, nucleosides, doxorubicin, different working like it is done in cancer therapy with combined drugs. And of course, we need very, very strongly to increase the cell entry efficacy. And we think that adding the delivery enhancers will be the best idea. So I will just thank you for your attention. And now the paper is open for questions. So I have seen that Domenica make a question. It says, could you give some comments on the kinetics of the self-assembly systems? Have you followed the information over the time? Is the standard annealing procedure applied to oligonucleotides sufficient in terms of times? Okay, so let's say that the formation is always, let's say, the same protocol. Is the annealing protocol that we use for making a duplex. So you just prepare, mix equimolar amounts of all the compounds, and then just heat and let it cool. It is true that with the DNA origami, the cooling is usually slow to make sure that the formation is quantitative. But in the DNA origami, in the DNA tetrahedron, sometimes it's better that the cooling is fast for some reason. The slow cooling, sometimes it led to some aggregation, but cooling fast is good. On the contrary, with the quadruplex, it has been described that the quadruplex have to be just prepare a very concentrated solution, and you let it heat and slowly cool, but then wait for 1 week even sometimes, because the kinetics of formation of the quadruplex is slow. But of course, one is formed, you just keep it on the refrigerator, and you can use it because it's always in the quadruplex form. What it could happen with the time and with the biological buffers is that slowly this quadruplex dissociates. So because of the kinetics, it is slow. The kinetics of formation. It is also true that the kinetics of melting is also very, very slow. So it can really work at least for 2 or 3 days in cell culture, and we don't see any loss of activity. So that is slightly bit special for each one. Then the second question is, thank you, Ramon, for the great talk using novel structure. Okay, thank you. You finally managed, because Mano asked me just one well in the middle of the Montserrat's talk whether I can give him the geometry, and it's good that you could enter. Thank you very much. So then there is in the chat. In the chat, we have also good things. I mean, Sven, thank you, Sven. So now all the speakers are already in place. So if you have still a question for each one of us, that will be great. Okay. Now we have a question from Mano. Yeah, the scalar map is the worst is the worst aspect of this story. Yeah, for the quadruplex and for the tetrahedron, this is not a problem because are a few oligonucleotides. So this could be scaled up as a normal oligonucleotide. The problem with the origami, the 300 oligos, okay, it is a lot of synthesis effort. And usually these so many oligonucleotides are made in a small scale synthesis. So this is one question, but I have to say that we use 50 nanomolar synthesis every time that we order, and it lasts for 3 or 4 years. So it is a big inversion, but then for the oligonucleotide side, I believe is not a big problem because it's only about 30 bases long, so they are not really complex oligonucleotides. The problem is the M13. The M13 is produced right now by just growing the how to say growing the bacteria, growing bacteria and growing the M13 virus, that is a bacteriophage. Is a bacteriophage, and you grow the bacteriophage with the bacteria, and then you harvest the virus, open the capsid, and purify the circular DNA. Of course, this is very expensive. What it does, most of the people is try to do some genetic engineering. And maybe not produce the same 700 bases long, but you could have some biotechnological production of this piece of DNA, maybe by genetic engineering. So that I will say that is what it will make this easy. We cannot do it because we are not experts in all this genetic engineering, but the Chinese group that made this nanorobot, for example, they made his own. It's not anymore M13, but it's something M13-like, let's say, that they can produce by genetic engineering. Then if this happens, this will be cheap and available to everybody. So we just need to learn some genetic engineering to produce a 7,000 DNA without virus. So that is it doesn't need to be circular really, but it is what is very important is to have it single-stranded, because then you don't have the complementary strand that disturbs you continuously. So this M13 bacteriophage is a phage that encapsulates his DNA naturally in single-stranded form. So, and that is you use nature to produce the chemical, no? But most probably, if the Chinese did it, it means that it can be done. Maybe making some double-stranded DNA first, and then melt it, and you get a nice. So the problem, in my feeling, is to be able to make a large preparation of this M13 DNA. Okay, we have a message from Ana Grandes that is the wife of Enrique, that she was all the. I also have seen the message from his son Danny. So we really miss Enrique. I know that you will miss much more than us, but we are really touched for his loss. Okay. If there is, okay, Anna, now we can close. I would say, no, we are well. We did not do bad. We more or less adjust to the time. We are only 15 minutes late, so it's not bad. So I will just close the webinar. Thank you for all of you for your attention. Thank you, MDPI and the journal Molecules for your support or organizing this event. And again, if you want to contribute to the special issue, you are more than welcome.
Yeah, thank you, Ramon, for sharing this event. I just shared through the chat a bit of information of if anyone is interested to have a certificate, you can write us to ask for it. I'm also sharing the link of the information of the special issue that it's on the conference on the website webinar website for anyone to want to contribute to it. Thank you to you, Ramon, Montserrat, Daniela, for your talks, for all the attendees for your attention, and that's it. I wish everyone a nice day, morning, evening, wherever you are in the world, and see you in a future event.
Okay.
Bye bye.
Bye bye.
Okay. Bye bye.